Which of these groups is not monophyletic? So monocots, seed plants, dicots, or gymnosperms? According to Campbell. Of course, according to me, I'd probably say these are not monophyletic, but it's not according to me. There's just a couple little questions here in the world. Okay, three more. Like, which one of these groups was not represented on that tree that I showed you? Is there one more? Is there one more? No? You lost somebody. There we go. Brother, that wasn't too good. Okay, so wasn't there a line that said monocots? Yeah, so that's not the answer. Wasn't there a line that says gymnosperms? So that's not the answer. There wasn't a line that said seed plants, but of course seed plants was what? The whole tree, right? Which was monophyletic. You could trace that back to a common ancestor. How about dicots? There was no line that said dicots, right? Instead it separated out Amborella and the magnolids and the star anise and all those things, right? Those all would have been considered dicots. Instead, there was a line that said what? Eudicots, but that's only a subset of what was historically considered dicots. Let's see if this is easier. The anthophyte hypothesis, y'all remember that? What does the anthophyte hypothesis say? It says that the Natales is potentially the sister group to flowering plants, right? So does it require that gymnosperms be non-monophyletic? Does it suggest double fertilization is a synapomorphy for the Natales and angiosperms? Does it seek to explain the origin of flowering plants? Or does it do all of those things? The anthophyte hypothesis. What does antho mean again? Flower. You're all going to remember that next week, right, when you're looking at anthozoa. You know, sometimes it gets better when we start animals because animals are a little more familiar to people. Yeah, we'll find out. You realize there's a whole set of names and tons of terms. So we'll see. Hopefully it'll be better.
Does it let you change your answer? Because it's like not letting you change it.
I don't know. Does it let you change your
Doesn't
answer?
it blink up once you change it?
I think it blinks. I see it blinking in here.
Oh,
There, yours just blinked.
I was looking at 18.
Okay, so are we ready? Let's see. Well, it's all of those. So if you're one of the 10 people who pick these, you're not wrong, right? But it's all of those, right? Gymnosperms have to be non-monophyletic because it says that the Natales are more closely related to angiosperms than they are to other gymnosperms, right? And it suggests this business of double fertilization that I told you is not the same thing, but that's part of the argument, right? And it also seeks to explain what the origin of flowering plants is. It talks about them being most closely related to Natalian gymnosperms. Okay, this should be easy. Which of these does not make wood? A palm tree, a banana tree, a bamboo tree, or all of those? Meaning that none of them make wood. Okay. So none of those make wood, right? They're all monocots. Yay, we're all done. Now it's just animals and ecology, and we're to the end of the course. Okay, so now, why don't we do this for about 20 minutes? Since we're going to have a chance to do it again on Thursday, right? And on top of everything else, the 1007 intro bio classes are running DNA fingerprinting right now. And they're doing it like with you, where you pour the gels and stuff. And like the instructor who's starting his lab at 7 o'clock has never done it before. So I'm going to try to help him, along with set up your lab practical. So, yeah, so I might want to eat something. So maybe we'll leave it like 6:40, is that okay? Anybody distraught over that? You guys are all gonna, those of you that have it tonight are gonna go somewhere and do something really enjoyable till 8:30, right? Go get a bottle of wine, assuming you're over 21, go get a bottle of wine, crack it open. Yeah, don't get too drunk before the practical, but do something so that you're nice and relaxed. And then whatever we don't finish right now, we'll do definitely on Thursday, and seriously, bring more questions, because if you stop asking questions, I'm going to be really tempted to start the animal stuff, because it'll help out so much this time. So you can either start here, or you can start with something else. Where do you want to start? Wow. Maybe you're ready to start the animal stuff right now. Come on, you guys come to me with questions all the time.
Number 1 was kind of tricky.
Number 1?
Yeah.
Okay, so I guess we're starting with number 1. So it talks about LUCA, right? What does LUCA stand for? Last universal common ancestor, right? And I put on here, it's a hypothetical organism. You know what that means, right? You realize all of these ancestors are hypothetical, right? Like presumably there's an ancestor here, there's an ancestor here, there's an ancestor here. Like if you ask me, I would say, well, mammals are a monophyletic group. What does that mean? Just in saying that mammals are a monophyletic group, that implies that I think that there is some common ancestor that all mammals share, and that all modern mammals are descended from that common ancestor. But if you ask me, who is that? I don't know, right? I'm assuming there's a common ancestor because I understand them to all be related to one another. Relationship means shared ancestry, so there must be a common ancestor. That doesn't mean that I have any idea who it is, right? You all understand that idea? So it's a hypothetical ancestor. It's not a real ancestor. Somebody in the other class said, well, what about Lucy? You all know Lucy, right? Australopithecus afarensis, right? Well, Lucy's not hypothetical, you realize. There's a fossil. Lucy's a real ancestor. Now, what she's an ancestor of, that's debatable, right? Whose ancestor is she? That you could debate. But she's not hypothetical, she's real, right? I mean, there's a fossil to support her. So do you all kind of get the idea between a real potential ancestor and a hypothetical ancestor? A hypothetical ancestor, we don't actually know who it is, right? We don't have no direct evidence to support a particular hypothetical ancestor. So it says here, you should realize that LUCA is a hypothetical organism. Presumably it existed, but there's no way to gather any direct evidence about it. Still, we might guess that LUCA was capable of reproduction via division, that it consisted of at least one cell, that it stored genetic information in the form of a DNA molecule. Why should we think such things? What indirect evidence leads you to those hypotheses about LUCA? How can you possibly know anything about it if there's no way to gather any kind of direct evidence about it at all?
Look at what every living organism shares.
Yeah. So this is supposedly the ancestor to all modern living things, right? All modern living things reproduce by division. All modern living things have at least one cell. All modern living things store genetic information in the form of a DNA molecule. So we might assume that the last common ancestor they had those traits, and that all of those traits have been inherited by all of the descendants of that ancestor. That's the assumption. It doesn't have to be right, but I mean, that's how you would start to guess that. You look at what, so like, for example, I told you I don't know anything about the last common ancestor for all mammals, but if you were to ask me, I'd say, well, it probably produced milk and mammary glands. It probably had an epiglottis. It probably had a diaphragm. It was probably endothermic. It probably had body hair that came from a follicle in the skin. Well, how could I know all those things about it without any direct evidence of what it was like at all? Because all modern mammals, all of its presumed descendants, share all of those traits. And I'm kind of assuming that they inherited those traits from the ancestor that they shared.
Do you think that it might have had two flagella?
Well, except, yeah, okay, so don't confuse LUCA with the last common ancestor for all eukaryotes. Yeah, the last common ancestor for eukaryotes, I don't know what that would be, Euca, I guess. Right? The eukaryote universal common ancestor or something. That would be reasonable then, because and I would also think that the last common ancestor for all eukaryotes had mitochondria, because all modern eukaryotes have mitochondria. right? So there's a whole bunch of hypotheses I could make. I would guess that it had complex internal membrane systems, things like endoplasmic reticulum and Golgi bodies. I would guess that it probably was unicellular. I mean, there's a lot of things I could guess about the last common ancestor that eukaryote shared, but remember, that's not Luca. Luca's going to be the last common ancestor for everybody. So you have to pick traits that every living thing, even bacteria, reproduce by division, have at least one cell, and store genetic information in DNA. You have to be picking characters that are true for every living thing. Okay, is that good? Could you all write out a good answer for that? Okay, so number 2, y'all know what heterocytes are? Not really. This is a heterocyte. The name just means different cell. This is Anabina, which is that cyanobacterium. It's symbiotic with Azolla. Remember I mentioned that briefly? Kind of, it lives in the roots of Azolla, that little fern that you looked at, that little water fern. So it says, describe the importance of heterocytes in the cyanobacterium Anabina. Be sure to talk about the problem associated with fixing nitrogen for photosynthetic organisms. Well, so if you fix nitrogen, which remember only prokaryotes are capable of fixing nitrogen, you use a set of enzymes to do it. And those enzymes, if you have free oxygen, they interfere with the functioning of those enzymes so that you can't fix nitrogen. This is a particularly big problem if you're photosynthetic, though, because if you're photosynthetic, you liberate oxygen as a waste product of photosynthesis, right? So here you are liberating oxygen, and yet you're trying to do something that if oxygen is present, it screws it up, right? So what they've done is they've compartmentalized the two different processes. They do nitrogen fixation inside of those heterocytes, which are thick walls, so that the oxygen doesn't get in there and screw up with the nitrogen fixation. And then they do photosynthesis in photosynthetic cells and other cells, so that the oxygen is not being produced in exactly the same place where the nitrogen fixation is occurring. Did you all get that down? In the other class, they didn't like that. They wanted me to write it all out. You want me to write it all down? Did you all get that? Did you use the word? I use the word compartmentalize. Right? The two different processes are compartmentalized. Let me write one so that you kind of see how you write this well. This other pin didn't erase very well at all. Are you even going to be able to read that? If I read over this? Can you guys even read that?
Yeah.
Free oxygen interferes with nitrogen fixation, right? Anabina compartmentalizes nitrogen fixation and specialized cells called heterocytes away from photosynthetic cells. Such that oxygen liberated from photosynthesis does not I guess I'll just say interfere with nitrogen fixation in the heterocyte, something like that. Doesn't have to be exactly like that, something like that would be an answer, right? Yeah, is that good? I'm assuming we're going through these in order. Is that right? Anybody want to skip number 3? Apparently not. Okay, 3 I like. So it says, the terms diploid, haploid, mitosis, and meiosis can never be used when talking about a prokaryote. Explain in as much detail as you can exactly why you can't use those terms when talking about prokaryotes like bacteria. And then it says, hint, show that you actually know what the words mean. So what do the words diploid and haploid mean? If a cell is diploid, what does that mean? Well, let's say the chromosomes are in pairs, they're in homologous pairs, right? What do bacteria look like as far as DNA is concerned? I mean, like if this is a bacterial cell, right, they have what?
One linear chromosome.
One circular,
Cir-
large
Oh, I thought it was linear.
DNA molecule. I would resist calling that a chromosome even, right? Some books do call it a chromosome. I'd call it something else. I'd call it like a naked loop of DNA or something like that. And then they have these smaller little loops, right, that are plasmids. They can build those little bridges and pass plasmids. That's kind of like how they have sex, right? How's that going to be? How are you going to have chromosomes in pairs here? It's not going to work. So the word diploid refers to a cell that has chromosomes in pairs. Bacteria never have chromosomes in pairs, right? The reason you have a cell that's diploid is because you have two complete genomes in that cell. One genome is maternal and one is paternal, right? And together you've had sexual reproduction by combining a complete maternal genome with a complete paternal genome into an individual. You have now an individual that has, in effect, two genomes, right? So that every chromosome exists in a paired state with another chromosome representative of the other parent. Bacteria don't even have sex that way. Bacteria just build a little bridge and pass a little plasmid and say, is that good for you, right? On they go. There's no combining, you don't have like two bacterial cells fused together like you do in eukaryotes. So you're never going to end up in a situation where you have two complete genomes combined into a single individual. They just don't have sex that way. Yeah? What does the word haploid mean? It means that the refers to a cell where the chromosomes are unpaired. Again, it wouldn't make any sense really to refer to these as haploid because there's never a situation where they're going to be paired, right? So how do you determine whether this is paired or unpaired? I guess you would argue that that's unpaired, but it's never going to be paired. So it's kind of a nonsensical thing to argue, yeah? How about mitosis and meiosis? Those words refer to what? Karyokinesis. They refer to nuclear division. Well, bacteria don't even have nuclei to begin with, so that's not going to work very well. And what was the definition I gave you for mitosis? Mitosis is when you start with what? Something that's diploid and you make more diploids, or you start with something that's haploid and you make more haploids. Well, if you don't even have haploids and diploids to begin with, how are you going to define mitosis? You can't define it that way, right? Likewise for meiosis. Just try to picture, you all know what mitosis is, right? You go through prophase, the nuclear membrane breaks down, right? Chromosomes condense, right? Remember all that? And you have like centrioles, you start to develop some kind of a spindle if it's an animal cell, right? Or you develop a spindle no matter what, but you have centrioles and develop a spindle if it's an animal cell, right? Then what happens in metaphase? All these chromosomes line up in an equatorial plane. How on earth is that going to work here? There's nothing like that here, right? And then you start to pull sister chromatids apart. There's just nothing like that. Try to picture this doing mitosis, going through those phases. There's nothing there to do that with. Yeah? So the short answers, you should definitely be telling me that diploid means that the chromosomes exist in pairs representing two complete genomes, one from two different parents, right? The fusion, in essence, of two haploid nuclei. There's no two haploid nothing that's going to fuse here, ever. That's not the way these guys, they don't have the type of sexual reproduction that would be necessary to have something like a diploid nucleus, right? So diploid and haploid refer to whether or not chromosomes are paired or unpaired within a nucleus, right? There is no pairing or unpairing here because there's not the same type of sexual reproduction where you combine the complete genome of two different parents into a single individual. And there's no mitosis or meiosis because those terms explicitly refer to nuclear division and either transitioning from a diploid state to additional diploid states or a haploid state to additional haploid states, or transitioning from diploid to haploid. But if you don't have diploid to haploid or diploid or haploid to begin with, there's no way that you can use words like mitosis and meiosis. Yeah? You all write out something kind of like that?
Won't they do like binary fission or something like that?
Yeah, you could call it that. So go ahead and call it that in bacteria, binary fission or something. Don't refer to, or you can say cell division. Just don't refer, technically mitosis, you realize, isn't really cell division. It's really nuclear division is what it is. So you can't refer to what bacteria do as mitosis. It's just not right. Cell division is fine, or binary fission is fine. Yeah. Okay, are we all good with 3? See why I like that? Because here I'm trying to get at, do you really understand what the words diploid, haploid, mitosis, meiosis mean? Okay, how about number 4? So 4 says, most fungal reproduction and dispersal, both sexual and asexual, is carried out via structures referred to as spores. What do all spores have in common? So what's the definition I gave you of a spore? It's not gonna get us into trouble. Notice if you're talking about a relatively limited group of organisms, like we're talking about only plants, you could say something like all spores are haploid, all spores come from meiosis, all spores are produced inside of sporangia, right? Because I'm talking only about plants, right? Spores are all produced by a sporophyte, right? That falls apart as soon as you go outside of plants, right? All of a sudden you need a much broader definition for spore, so you're not going to be able to say all that about spores. So what can you say about all spores? What makes a spore a spore?
Single cell.
Okay, so spores, as far as I'm using this word, is always a single cell. And I don't really think that there's... So it's a single cell to start out with. That makes things like Jimmy are not spores, right? Because they're multicellular. And then the second part of that, to distinguish it from a gamete, which is also a reproductive single cell. Spores, what?
Doesn't need to fuse with another
Yes.
cell.
Do not fuse, or, sorry, let me change it. Let me say develop without fusing. That's about it, right? Otherwise, there's going to be, especially within fungi, we'll use it to sort of, because the second part of this says, you know, describe the differences between zygospores, conidia, ascospores, and basidiospores. You'll see there's spores that are sexual, there's spores that are asexual, there's spores that arise from mitosis, there's spores that arise from meiosis, there's spores that are haploid, there's spores that are diploid, there's spores that are endogenous, there's spores that are exogenous, and there's even a spore that arises from karyotomy, right? So all bets are off now, but in every case, they're a single cell and they develop without fusing, right? So that's going to be universal. But so let's see, if you just tried to do those, what are they? Zygo, conidia, basidia, and ascospores, right? So maybe you can even draw a little picture of each one. Here's a zygospore, right? Conidia, maybe something like Aspergillus. Right, pretty little. Basidia, little club, typically with 4 spores on the outside. And an ascus would be what? Little sac, like Beziza, right? Okay, so how about, let's go diploid, haploid. So what can you say about a zygospore? Diploid, conidia are haploid, basidiospores are haploid, ascospores are haploid. How about, what could you say about whether they're sexual or asexual? Zygospores would be?
Asexual and sexual.
Well, no, but not the zygospore. The sporangium is asexual, but the zygospore is sexual. And then the conidia would be what? Asexual. And these are coming from a meiotic division of the zygote, so they're considered to be sexual. Right? Okay, how about whether or not they arise from mitosis or meiosis? How about that? So let's skip the zygospore. How does the zygospore arise? It doesn't really arise through a division, it arises through karyotomy, right? That's the origin of it, right? How about conidia arise from what? Mitosis or meiosis? Mitosis. And basidia and ascospores? Basidiospores and ascospores arise from meiosis. Right? Remember, this is that zygotic meiosis where the zygote is the only diploid part that does meiosis and make typically spores. Okay, and how about if we could put whether it's endogenous or exogenous? That doesn't really apply to a zygospore, but how about like conidia? Those would be what? Exogenous. Basidiospores would be exogenous. Ascospores would be endogenous, right? That kind of separates them out, right? There's probably other things you could put too, but I guess you could write a little sentence here. You could write something like these spores are produced inside of a sac structure. These spores are produced on the top of a club structure. Okay, let's do one more, then we'll pull out for today. Okay, so this says the old protozoan phylum Sarcodina included all of the protists that moved with pseudopodia, right, with a false foot. So that would have been things like foraminifers, radiolarians, or arachnophytes,
All the Rhizarians, right?
all the rhizarians, plus the amoeba, right, like amoeba proteus, the one that you looked
That's
at.
the loop pseudopodia though, that's converted.
With the lobe pseudopodia, well, yeah, hang on, to the end of the question. Hang on to that
Oh.
part about being convergent. And what else? Also things like dictyosteliid slime molds. Basically, anybody that moved with pseudopodia would have probably been thrown at one point with sarcopenia, although slime molds might have gone into fungal like protists or something. So there is no such group in the current classification. What happened to them? Okay, well, we're kind of saying it now. Some of them with the thread-like pseudopodia went into the rhizaria. The rest of them are classified as unicons, at least in Campbell, under the amoeba zones, right? And then it says, what can you say about the evolution of pseudopodia in eukaryotes, right? So there's a lot of cells that are capable of phagocytosis that have in effect pseudopodia. You have them in your own body, right? You have lymphocytes and things that are capable of actually engulfing like bacteria and destroying them, right? They have, in essence, pseudopodia that are capable of phagocytosis. So you in your own body have cells that are amoeboid in a sense, right? So what can you say about the evolution of pseudopodia? Clearly, pseudopodia are all over the place if you look at eukaryotes, right? If you had a eukaryotic tree, right? I just answered this the other day with like Joel or something. Didn't you ask this, Joel? I can't remember why. Because it was in the lab manual or something too. So if this is the tree, and here you have what? Maybe pseudopodia, something with pseudopodia, here's something with pseudopodia, here's something with pseudopodia, and here's something with pseudopodia, right? So clearly this cannot be synapomorphic. It cannot be something that evolved only once, right? If you have it only there and not any of the other branches. So the question is, what are the two possible scenarios to explain the evolution of this? What does it say? It says, what can you say about the evolution of pseudopodia in eukaryotes? There are two possible scenarios. Okay, so what are the two possible scenarios? One is that the trait is, what did you say, Joel?
Convergent.
Convergent, meaning that pseudopodia evolved here, pseudopodia evolved independently here, pseudopodia evolved independently there, and pseudopodia evolved independently there. So each place I see pseudopodia, it's a convergent trait, that pseudopodia did not evolve just one time. It couldn't have, because where would it evolve just one? Well, it could have. That's going to be the other scenario. But you'll notice in this particular scenario, it's evolving multiple times. Okay, so what is the scenario where it could have evolved only once? How can you end up with pseudopodia evolving only once and yet end up with it in these unrelated lineages? It's not a convergent trait. It's not derived at all, but it's actually what? The ancestral condition. So you could say, oh, no, pseudopodia evolved just one time. It evolved way back here, right? So pseudopodia was present there, and these guys are representing that ancestral trait still. But guess what? Pseudopodia were lost here, they were lost here, lost there, lost here, lost there, lost there, lost there. And with all those little losses, these guys are just exhibiting that ancestral trait. Yeah? This is why any group that's defined by a convergent trait or by an ancestral trait is not going to be monophyletic, right? There is no branch leading to some common ancestor that they share where you can have this thing evolve one time and be inherited by all descendants of that ancestor, because they're not a related group. It's an unrelated group, the things that have pseudopodia. They don't all form a natural group or a monophyletic group. So there's only two possible scenarios, or it could be a combination of the two, right? So for example, you could have some convergence. You could have had pseudopodia evolve here and here, or something like that, and then you had some losses, right? So it's either a combination of being slightly a shared ancestral trait, and also there's some convergent origins. In all likelihood, pseudopodia is probably a convergent trait across those groups that I just mentioned, right? Or it's completely convergent, or it's completely ancestral. But I'm going to ask you that question. You know I'm going to ask you that question, right? Like I'm going to say, for example, let's think of something else. Photosynthetic ability is another one that would be really easy. So photosynthetic ability in eukaryotes is found across a bunch of different unrelated groups, right? Some straminopiles, some of the alveolids, right? Some of the archaeplastids, most of the archaeplastids, right? All these different groups that are unrelated to one another have photosynthetic ability. So what are the two possible evolutionary scenarios for photosynthetic ability in eukaryotes? It's going to be what? That it was either acquired multiple different independent times that it's a convergent trait or it's a homoplastic trait, or that it represents the shared ancestral condition, right? And a lot of different individuals have lost it, right? Such that the only individuals that share it, they're just exhibiting an ancestral trait, not a trait that's derived for them. Yeah. In the case of chloroplasts, it's probably the former, right? It's the idea that because you have all of these different primary and secondary acquisition events, that it actually represents a bunch of different photosynthetic ability acquisitions, but not the ancestral conditions. Okay, so I'm never going to remember because I ended in a different place with the other class. You'll remember which questions we were on, right? Seriously, there's no way it's going to take an hour and 20 minutes to go through like 3 questions up here. So if you don't want me to start talking about animals, you got to bring something else to talk about. Right? Either that